Hi everyone. Hello and welcome again. Microbehunter here. Welcome again to another Saturday live stream. And yeah, I'm very happy that you could join me again today. As always, I'm trying to get the setup correct. Yeah, okay. I think everything seems to be working. I already see that some people already joined the chat. Yeah, hello all around the world. Today, yeah, I want to do again a little bit of a more traditional microscopy. Nothing totally exciting, or maybe a little bit exciting, I don't know. We're going to see, because I would like to put some moss specimens under the microscope. Well, not so much the moss itself, but rather the microorganisms that we find on here. Why? You might wonder. Well, yeah, I have to be honest with you, the last couple of weeks, I've been putting sand samples under the microscope, mostly non-living things, some polarization microscopy. It's actually one of my boys who then recommended this. Is, yeah, over the last couple of weeks, you've only been looking at sand. Don't you think it's again time to look at a few living specimens again? And said, yeah, actually, it's a good idea. And for this reason, I would like to put some moss today under the microscope. And I know that there are a few tardigrades in here as well, but not only. So we're going to have a little look here. Yeah, as always, I would like to say hello all around the world. If you have any microscopy-related questions, even if they're not specifically, even if they're a little bit off topic, as long as they're microscopy related or biology related, I will try to answer them. But please do indicate this in the chat by putting an @Microbehunter or @Oliver in front of the message, so that it's easier for me to find the text, because I'm not able to read all of the text messages. I cannot multitask. I cannot talk at the same time and read at the same time so easily. So for this reason, I'm going to simply skip over. Yeah, and I can see that the sound is good. Thank you very much. Okay, the first 3, 4, 5 minutes, I usually go through all of the hello, so that basically to give people some time to join in. Yeah, so hello from Belgium, from Vietnam, from Montreal, from Vietnam, okay, from Northern Germany, okay, yeah, from Nordrhein-Westfalen, from Germany again, California, United States, okay, yeah, sound is good, thank you very much. Okay, and for the first question, okay, yeah, I'm curious to see if there will be a difference from what's inside the moss versus mine in Canada. If there are no differences, I will ask you to explain why. Well, I will tell you, because there is a difference between those two type of moss samples here, because I think it does not depend so much from which country the moss is from, but from the highly specific place where you collected the moss. And that's actually one of the things I would like to do today, because this moss here on the left side was originally very dry, from a very dry area, and this one was from a very moist area, and it's quite different. Okay, it's not so much, I think, not so much a question of which part of the world you collected the moss from. This might also be a factor, but I think the actual place where you collected the moss from can make a huge difference, and that's actually something I would like to talk about today as well. Okay, yeah, okay, San Francisco Bay Area. Also as well, India, very nice. Okay, so what I'm going to do is to tell you a little bit of some background here. Okay, if you're into hobby microscopy, and I guess pretty much all of you are in a certain way or another, one of the most common things to look at are so-called water samples. Okay, so you collect some pond water, usually fresh water, usually some sediment from the bottom of the pond, and then you put it under the microscope, and you're going to see a lot of ciliates and rotifers and single-celled organisms and whatever. Yeah, but you don't have to always look at pond water. Moss is a can be a very, very rich source of interesting specimens as well. Okay. And what I'm going to do now is I'm going to, by the way, I'm in the process now also of making a video on how I collected those moss samples. So if you check this channel in maybe today after the live stream or tomorrow, I'll upload a video, okay? Which talks a little bit more about this as well. But generally what I would like to say is that those two mosses have been collected from very different places, okay? So this one over here was collected right outside my apartment from a very dry area. So the moss originally was relatively dry, and I added a little bit of water. I've done this several hours ago. And this one over here was collected from a very moist area, and we're going to see that the biodiversity is quite different. Okay, so what you do is the following. You take your moss sample, okay, and I'm going to first show you the moss sample where there is little, okay, to see. Maybe more than a few hours ago, because a few hours have passed and the microorganisms already started to regenerate, okay? But what you do is the following. You add some water, okay, like this, okay, and then the fun starts really, because all you do is that's what I usually do, is you take your fingers, yeah, and you simply dip the moss, yeah. You also can turn it around, yeah. You see there's a little bit of soil here, and what you're gonna do is you're going to kind of rinse out and wash out, yeah, some particles, microorganisms, whatever you can find in here, okay. So you can be generous with the water a little bit, but what you really want to see is you want to see those particles here, okay? Some decomposing leaf matter, some soil particles, of course, some microorganisms. And what you want to do is yeah, you do this for a couple of seconds, just like I've done, okay? And then you just leave it like this for a minute or so. I'm just looking for a place, for something to kind of hold it up. I'm going to use this cap, and you simply allow the all of the particles to settle down now. Okay, and this is going to take a minute or so. Yeah, also some microbes that are floating around might also start to slowly settle down to the bottom of the Petri dish, and we're just going to wait now. And I'm going to use this time again to do a little bit of reading here. Okay, hi from Switzerland, from Texas. Okay. I cut my hair myself. Somebody mentioned that I cut my hair. Yeah, and we're going to look for tardigrades as well, okay? So, and yes, most of what I find in the moss each time are nematodes, ciliates and rotifers, but so far there has only been one tardigrade. Let's see what I'm doing wrong. Well, I think you're not doing anything wrong really. What I found out is that it's not really so much a question of getting them out of the moss, but it's simply more of a question of where did you collect the moss from. So I have found vastly different moss samples, okay? I remember a few years ago I collected a moss sample from a forest, actually a dry one. Yeah, and I added some water, and I found just on one slide, I found 6 tardigrades on one slide in one tiny drop of water, right? And then you find other moss samples where there is nothing. Yeah, so I would say it probably is not really worth trying to find something interesting in your sample. Just try a different moss. Okay, that's almost my suggestion, right? So we're gonna, but I know that in the other sample that I have, there are a few tardigrades. Not so many, but I found a few. Okay. I think the dry moss will have more tardigrades. Well, the thing is, it is correct that the tardigrades are basically are able to survive dryness. That's correct. So when the moss dries out, then the tardigrades will go into a state called cryptobiosis, which is a very a dormant state, and they're able to survive this way. However, it does take some time for them to come back to life once you added water, and in dry moss I have not always found so many living things. Okay, so we're going to give it a try. So this should be now the sample, which is kind of disappointing. Okay, so don't be disappointed. Yeah, because this one here, I'm using to simply show you how it looks like when there is not a lot to see. I mean, a little bit maybe, but not really a lot. Okay, so this is what I would like to do now. So yeah, I'm going to do the following. Before I do that, I'm going to explain the pipette here that I'm using, because has also been a question a little bit. I'm using a microliter pipette. I mean, many of you probably know disposable ones like this. That's also fine. I switched over to those because it's for convenience. Yeah, this one wasn't too expensive. By the way, not sponsored or anything. I just bought this some months ago myself, right? And what you can do is by turning this here in the back, you're able to change the volume. So I'm currently set it to the highest volume, 50 microliters. And what you do is by pressing in, you're able to take up liquid. Yeah, and by pressing this button over here, you're able to eject the tip. So what you do is you put a tip, the disposable tip on here, right? And when you press this button over here, okay, then this will move forward and it will kind of eject it. And so basically you can quickly exchange the tip. Yeah, and by pressing this here, you're able to take up liquid. Yeah, so that's what I'm going to do now. Yeah, and what you want to do is you want to not just take the liquid, but you want to take the solid material, right? So you go all the way down now. Yeah, and you try to take the solid material, and then you're going to discover that sometimes this works and sometimes this doesn't work because sometimes it becomes clogged. Right? So what I'm going to do now is, it didn't get clogged now, but of course what you can do is, I'm just going to show you, because it's also something that I'm doing quite frequently, to prevent clogging, or yeah, you'd simply take scissors and you cut off the tip a little bit. Okay? And if you do that, yeah, of course it becomes wider, but you're still going to take up the same amount of liquid. Okay? Yeah, so I'm just, this was just more or less a kind of a demonstration, right? What happens now is that you're able to see this maybe a little bit, and then usually what I do is I hold the pipette vertically to allow the things to settle down again in the tip, because sometimes I have more liquid in here than I actually want to have. Yeah, and then I simply put it on a microscope slide, and then you see that there is still a small amount left over in the tip, right? And in order to get that out, you press this completely. So it's basically, it's only the first stop here, but I'm able to press it down further, and then it kind of throws out the rest. I just show you here over here. I'm able to push it down yet further, and this kind of ejects the rest. And then with this sample on the slide, I can now throw this tip away. Actually, there is no reason to throw it away because I can just reuse it. If you do not want to transfer any contaminations, what you do is the following: you simply press this button here, right? And then look, I'm going to catch it now. Yeah, it's ejected. Yeah, and what I usually do is I kind of inject the whole thing into a little trash can. Yeah, this is my trash can, and then I do the following. Yeah, and gone it is. Okay, so that's simply I wanted to show you, and then you normally put a cover glass on top here. I will also do that, and I of course misplaced my cover glasses. No, they're hidden behind here. Yeah, and again, if you just joined in, do not be disappointed with this one over here, because that is actually the sample where we should see few specimens. Okay, so this is just a little bit of a reference here, and let's put this on here. Of course, this, yeah, and you see that, you see that it's not completely flat because of the soil particles and so on. Yeah, but it's fine. Yeah. The question is now, will it damage the pipette if water comes into it? Well, generally, any water should not be coming into the pipette itself, because it won't allow me to suck up water all the way. Yeah, but we used in the lab. We used pipettes like this, more expensive one. This was pretty cheap. This was ridiculously cheap, actually. I think around 20 or 30 euros only. Normally, the good ones, the quality ones, cost hundreds of euros. I don't need that precision that they have, and they're calibrated because they're really used for research. This was just it's a conventional one, so to say, but it won't allow you to take up water all the way. I'm just going to illustrate this, right? Because this is now the maximum volume that I have, and if I do have a water sample somewhere, no, I'm just going to take up more of this. And look, if I pull up, yeah, it will go only up to here. Yeah, there is no way for me to suck it up all the way. Yeah, so, and yeah. And off it is. And I'm going to now show you what else. I'm going to now put this under the microscope. And where is the scope? That's the microscope view. Now you can also see the magnification. Yeah, and you see the thing here. And let's put it up. And this is basically what we see. Okay, I can actually flip this over. There is, I cannot believe this. Do you actually see that there are a few things, tiny things moving around? This a few hours ago this was not visible. I will tell you why, because it was a dry sample and it simply took some time for the microbes to regenerate. But essentially, where is the arrow again? Here it is. Yeah, if you look carefully, there is some tiny things moving around. Okay, but honestly, most of the things are not really super exciting here. But let's go up a little bit with the magnification. Just want to show you. Yeah. It's a little too dark. We have to refocus again. We see a lot of particles here, soil particles, but you get the idea. Yeah, a little bit maybe, but yeah. And so usually what I do is I scan the slide like this. I get an overview, and if I do not see a lot of movement, then essentially I say, bad sample. So then generally more interesting specimens like tardigrades and so on are also generally not so easily visible. So this is, so occasionally I saw some so-called amoeba tests. These are the shells of certain amoeba. I don't know what this round thing here is. There was a little bit of movement here. You know what this is? Yeah. Hidden behind a soil particle, it's a mite. Yeah, they can also be found in moss. Yeah, so somehow this one survived the dryness without problems, but those mites are quite frequent as well. Yeah. So I'm going to, as we adore the mite, or are the two of them? I don't know. I'm going to look again here, a little bit some of the comments here. Yes, let me quick. Most of what I find in the moss each time are nematodes. Okay, yeah, I already read that. Okay, where can I get those Petri dishes? Are they local shops? I know I could buy it online, but it's always nicer to inspect the hardware in person. Let's put it this way: Petri dishes and so on, they're kind of specific, so you will not find them so easily locally, I think. So I usually, you can buy them over Amazon, for example. Honestly, you can also get plastic ones, but the plastic ones are usually disposable ones. But I probably would get glass ones because they can be easily cleaned. You can put them into the dishwasher as well. Yeah. That's usually the thing that I recommend. Yeah. And I collect dry moss from the same trees in a local oak woodland forest and almost always find a few tardigrades. Okay? That's cool. They're in a ton state and rehydrate quickly. The ton state is when they basically are in the state when they are dormant because of dryness. Okay? So I got one of those micropipettes on Amazon. Yeah, cheap and easy to use. Again, there are hugely expensive ones as well. Yeah. This was the question with the water. Okay, so that, but essentially go down with the magnification to get a better overview again here. But I think you do get the idea a little bit that not so much. Okay. Yeah, you might find occasionally something, especially. So my suggestion is if you have a moss sample like this, add a little bit of water and let it stand for a few hours. And then you're going to see what develops, right? Of course, certain microorganisms that were able to survive the dryness, they will start reproducing. And that's what we've seen here. Yeah, but bacteria are going to probably reproduce as well because some of them will have survived, or a lot of them will have survived the dryness. Yeah. So, but generally, I think we all agree that I don't know what this is. Generally, we all agree that there's probably not so much here. Okay. Is there a solution to slow down an organism in? Let me find this again. I missed it. There was a question about what can you do if an organism moves too quickly, and I lost it. So there was somebody asked is there a way that you can are able to slow down? Is there a solution to slow down organisms when they are too fast to study? Yes, there is a commercial solution called ProtoSlow. I have only used it once a long time ago. ProtoSlow, SLO, it's called. It's a trademarked thing, which is a solution that you put over your sample and it slows down protozoans. However, I think it's not necessary, because what you do is you take a very little amount of water and make sure that there are no soil particles inside on your slide, and then you kind of squeeze or sandwich the paramecium or whatever protozoan you have between the cover glass and the microscope slide. This will press them flat a little bit, so you're able to see some of the cell organelles even a little bit better because of a better depth of field that you have, but it will also slow them down. Okay, so the first thing that I would try is I would actually try to use very little water, so that to also limit the vertical movement, because if organisms move vertically, then they will move in and out of focus, and that's really difficult to chase. Yeah? But I have a long time ago at university time we've used a ProtoSlow, which is a trademarked substance that can be used as well. Okay. So the next question, which is important, normal tap water. Yes, that's something I wanted to talk about. Yes, in my case, this is normal tap water because it is not chlorinated. Okay, so the place where I live, it is not chlorinated. So it has drinking water quality as well. And you do not want to use distilled water either. Because sometimes I read somewhere you add distilled water. No, you don't want to do that because distilled water does not contain any minerals. And it might, it's not osmotically, from the sake of osmosis, it's not good if you add that. I will contradict myself now. Is this because you put it over the moss? It's not going to be distilled anymore, right? But generally, normal tap water will work. If you don't have non-chlorinated tap water, you just use bottled drinking water or mineral water without gas, without carbon dioxide, okay? Yeah, wallpaper paste, that's correct. I said wallpaper paste is some kind of a some kind of a cellulose type of thing that I read. I've also heard that you can do that as well. I've never tried that. Okay. So methyl cellulose. Yes, methyl cellulose. It's the thing. So maybe it's the same in ProtoSlow. So what I'm going to be doing now is I'm going to, you get the idea. So what I'll be doing is I'll be ignoring this moss sample now because there is barely anything interesting to see. Okay. So let's use the different moss sample, and I'm going to switch back now to the desk view. Here we go. So this one I'm going to put away. And this one I collected a couple of days ago. Again, the full video will follow where I collected this. And I was kind of surprised because a couple of days ago, this thing was, I've rarely seen a moss sample with so many microbes in here. So, I hope that this is going to be again the case. Okay. And the difference again is that this moss grew at a place which was permanently moist. Yes, so this was actually growing on a wall, but I could actually see that there was a lot of moisture coming out of the wall because of for this reason there was of course plenty of time for microorganisms and tardigrades and whatnot. Yeah. To kind of grow and develop. And I actually realized that this is probably good moss because there were fern plants growing right next to it. And fern is actually a sign that the moisture is high, because otherwise the fern won't grow. And so I said, well, if fern is able to grow there, then probably the moss is also able to grow quite well. Okay, so that's a little bit, yeah. I'm going to leave it now like this. I'm going to give it again a minute or so, for the whole thing to kind of settle down a little bit. And then we're going to give it a try. And I really hope that this one is going to be as successful now as it used to be a day or two ago. Okay. Same slime they used for the special effects in the movie Alien. Oh, I see. Okay. Yeah, and sometimes in the movies they're using the slime, right? And that's apparently methyl cellulose, which is also used as to make the things viscous. The thing is the following: when you add something to microorganisms, you do not want to change the osmotic or the potential around too much. Yeah, you don't want that the organisms lose too much water or gain too much water. So it's got to be kind of neutral a little bit. Yeah. So this is the thing. Yeah. I don't know which type of moss it is. I'll be honest with you. Okay, I wouldn't even be surprised if these are different species together, right? Yeah. So what I'll be doing here, I see that the particles here are pretty large again. So I'll be using again my scissor. Don't I have bigger scissors? Where are they? No, here they are. Here they are. So I'll be again. Let's do it like this. Up. Oh, this was not good. Okay, doesn't matter. This, of course. So and yeah, let's take some of the solid material again. I usually wait again for the particles to settle down a little bit. I don't know. Let's try it again. Because then if you let them settle down, then you do not have to use all of the liquid. Who cares? We're just going to try it like this. And I usually wipe the cover glasses, and I really hope that this is going to be now as successful as it used to be. I really made sure that over the last couple of days, ever since I collected the moss, it doesn't dry out, so I always kept it moist. Up. And let's give it a try. It's maybe a little bit too much liquid. Oh, it doesn't matter. I'm going to give it a try. Okay. Methyl cellulose powder. So basically, this seems to be like a new suggestion for a new video. This is to actually make this substance yourself so that it slows down microorganisms. Scope view. Yes, it's not centered. And I really hope that there is more movement going on here. Actually, I'm still a little bit disappointed because there was so much more going on. Oh yeah, there's a little bit more going on. Actually, here it is. Here is more going on. Okay. Yeah, here. Yeah, so we're able, you know what, I'm going to go up with the magnification already. And we can already see a range of assortment of different, single-celled organisms as well. I've seen a rotifer already. Here, this one is a rotifer. Yeah, the one in the center. Yeah. A nematode worm is over here. What's this here? Seem to be also two ciliates floating around. Here as well. So you see there are plenty of things to explore here. What is this here? That's a huge nematode worm. Yeah. Our little friend again. Yeah. Our mite. Yeah. This guy is not able to move forward. I think it's so tall that it's now sandwiched between the cover glass and the slide. So it's kind of, the legs are kind of in not able to make contact. Yeah. So what else do we have? Yeah, and that's basically the thing that I wanted to show you as a little bit as a proof of concept that if you're not successful finding something in moss, change the moss sample. What in the world is this here? It almost looks like the head of some kind of an insect here in the middle. Yeah, so especially if you make a video and you do some how do you call this, a time lapse, yeah, then you're able to see a lot of movement. Yeah? And what I actually wanted to do to illustrate to you is if I find a tardigrade, that I actually use the pipette to take it off the slide and transfer it to a different slide where there are less particles. What is this here? It seems to be some kind of a crustacean or I don't know, an arthropod in any case. Okay. Yeah, so, yeah. So this sample is quite generally quite satisfactory, I think. Normally I've never, in moss I have usually never seen so many of them. What you can, this is a huge nematode. Of course you can try to make enrichment cultures as well, and this basically means that you'll add a little bit of food to the sample, and then you kind of hope that whatever microorganisms are in there that will start to reproduce. Okay, so this here, what you see right now in the center seems to be like the capsule that has popped open with the spores in there. So, and this here in the middle now seems to be the exoskeleton of And this kind of illustrates a little bit to like one of the viewers has said, is they move around so quickly that it's kind of difficult to actually chase them around. Yeah. And in this case, you want to add probably this methyl cellulose, which I think I'm going to start experimenting. Oh, is this lacrimaria? But that would be cool. You see, I'm losing the focus. Yes, that's. I think that's lacrimaria. This is one of those that they have this huge extensible neck. I think it might be. It's a ciliate that has this long extension, but we have to see if it's able to really extend it very long. Yeah, because it uses this long extension, like it's almost like a trunk, to hunt. Yeah. But you see, it always goes in and out of focus because it's too thick. Yeah, the sample. Yeah, because of all of the sand grains and the leaves and so on, they kind of prevent the cover glass from actually coming down further. All right. So I'm going to now go back again to the questions. A biology question here. Can you explain ciliates? Are they prokaryotes or eukaryotes? They are eukaryotes. They're protists. And they are single-celled eukaryotes. And they're called ciliates because they have tiny little hair on the surface. They're so-called the cilia, and they use those cilia to move around. You also have so-called flagellates, and they are also protists. Protists are also eukaryotes, and they have generally one long whip-like extension that they use like a propeller to move forward. Yeah. So ciliates are eukaryotes. So their cell structure is similar to our cell structure. I know it's kind of weird when I say that, because we think that they're actually we're so different, but actually on a biological level, there is a lot of similarity between living things. Yeah. So I'm going to quickly move over again to the questions here. I'm surprised that the mite didn't get squashed by the glass. This does happen sometimes. Okay. Here's the answer. They're unicellular eukaryotes. Here we got the answer. Yeah. My microscope takes pictures and videos, but always loses focus and depth of field. Do I need to tighten something or anyone? Generally, if your microscope loses focus, be careful. It's not that the specimen moves in and out of focus, but it is focused, and then all of a sudden everything starts to become blurry, right? And then if this happens, then your stage goes down on by its own weight, and then you have to tighten. The coarse adjustment knob, and there are ways to do that. Depends from microscope to microscope, but you can actually adjust how tight the stage moves up and down. Okay. So if the whole thing goes out of focus, then the stage sinks downwards. Yeah? So which food did I add to the culture? To this culture, I did not add any food. because this is just as it came out from the moss. But if you want to make an enrichment culture, generally what you can do is you can add a crushed wheat grain. They are quite recommended. So you crush one between 2 spoons and you add it. I can show you how they look like. Yeah, and I've got something else that I want to. Very quickly, I'm going to switch over to the desk view just because somebody asked. So what I've got here is these are wheat grains, and what you do is you crush them between 2 spoons and you drop it into the water sample. The white little tiny, that's dry yeast, and this is a new thing that I bought. That's algae. They sell this as a food supplement. These are chlorella algae, and I don't know why people would swallow those. I bought this also over Amazon, but I've scratched off some of the things here because I'm using this as a food source for my water fleas as well. I don't even know where I don't even know where I put the here it is. Just a second, I show you. Yeah, here it is. So basically, I bought myself those chlorella tablets. It's again not sponsored or anything. I just bought this myself as a food supplement. Yeah, and the reason is because I wanted to because tardigrades actually feed on these chlorella algae, and I thought maybe I could kind of grow some tardigrades. This was actually the project. I didn't start yet. Okay, so this is so just let's go back again to the scope view here. But I have no tardigrades so far. Okay. Yeah, there are probably prokaryotes all over the slide. No, definitely. There are lots of bacteria probably here. I can have a look later on a little bit, but to see bacteria, I have to make sure that there are not too many particles in there. Okay? Otherwise, there is the bacteria will go out of focus. I could actually try some phase contrast later. But a little bit disappointing, at least right now, is the fact that I did not find any tardigrades yet. So let's go down with the magnification. Let's move away the filter. So this is not the lowest. Yeah. And usually, here is our mite again. I just want to say that usually what you can do is because tardigrades very often like to adhere to soil particles, and they like to cling to them. So sometimes if you look at soil particles, then sometimes this is a place where you can also find them. Okay, so for example, yeah, this one here in the center, yeah, it seems to be a rotifer. Okay, maybe I'm just going to show you again using the arrow. This one over here, it's too long for tardigrade. Yeah, but these moving soil particles or debris or whatever it is usually is a good place also to search for tardigrades. But again, a little bit unfortunate that at least here I'm not able to find any yet. But this doesn't mean that there are none present. It could be that essentially I overlooked them. Just for your info, in the previous trials that I made with this moss sample, I usually found 1 to 2 tardigrades per slide after some searching. Okay, so after some searching. Yeah. So I was surprised how good water samples look under the stereo microscope. Yes. I also have a stereo microscope, and however, it is good for different things, right? So if you want to look at water samples under the stereo microscope, I actually recommend that you take maybe some leaf of a water plant, and try to observe maybe the biofilm that is growing on the surface. Look at this soil particle in the center right now, right? It was actually moving. So sometimes, or these large soil particles here. Yeah. You always have to check a little bit if. See, something seems to be hiding here, but I don't know what it is. Yeah, it's difficult to see. No, it's too. Is it too? Yes, I think it's one. This could be a tardigrade. Okay, where? Look here. behind here. Okay, I think I saw several legs. Okay, here this guy over here could be one. Not this one up here, but the one that's hiding here behind the soil particle. Yeah. So, yeah. Let's see. Yes, yeah, it's pretty. I'm almost sure it is one. Yes, it's a tardigrade, definitely. Okay, so that's not the thing. The problem that we have now is we found one, but this guy is yeah, clinging to this huge thing here, right? But I want to see it better. Okay. So this is a little bit the problem, and I'm going to try now something, and just for the sake of demonstration. So, yeah, it's a tardigrade. Okay, so it's not going to run away. It's going to stay on the soil particle because that's what it likes to do. Oh, dang! I cannot do that. What I wanted to show you, because I put a cover glass on my slide. Dang. Okay. So this is a little bit something where I would like to, I have to redo the slide, because what I wanted to do is the following. I wanted to use the pipette here to actually take off the tardigrade and transfer it to a different slide with cleaner water on it. Okay, so that I can get a better picture, yeah, a better view of it, all right? So I cannot do that with the cover glass on top. Okay, so but I will make a new slide without a cover glass, and we're going to try to find another tardigrade. Yeah. But I think but for me, what is important for me to illustrate is kind of the proof of concept that yeah, they can be found with a little bit of patience. Yeah. So if I lift the cover glass, that's of course also something that I tried to do, is then I'm going to lose it again. It's going to be somewhere, right? Yeah, so it's probably easier to simply make a new slide and not put a cover glass on top. Okay, so that's what I'll be trying now. Okay, yeah. So that's you know what? Let's give it a little color. See, that's I've switched on DIC now. Yeah, and you can, if you want, you can add some color as well. Okay. Yeah. In case you really want to see some nice tardigrade videos, I am currently editing one, which will be uploaded very soon. Yeah. So, but you know what? Look how strong it is. It's kind of pulling on the whole soil particle. You know what I'm going to do is I'm just going to redo the whole thing, and this time without cover glass. And maybe if we do not find the tardigrade, then I'll try to take some other organism. So desk view, here we go again. Yeah, we see now that the whole thing has settled down now, hopefully. Actually, look, I just want to show you something here, and I'm also going to tell you why I'm probably not going to use that. It is also possible, and I've also shown it to you. If you look carefully, this is a slide that has is concave. It has this indentation in here, right? It's suitable for holding water droplets. But the thing is if you use those, then the droplet can be quite thick, and then again the organisms move up and down, right? So generally, I will sometimes use those. Only under special conditions, if I really want to make sure that there is a lot of space, between. But otherwise, I'll just use the regular microscope slides. Okay, let's move this out of the way, and let's simply try this again. Again, I will try to collect, and in this case, I'm going to really, because I know that the tardigrades really like to cling to particles, I'm going to try to, if possible, only put the particles on here. So I'm going to really wait a little bit for everything to sink down a little bit, and it's not very, I want to get more. See, it's still not able interesting. The interesting thing is it's still not able to take up the larger particles, and I wonder why. It could be maybe when I cut it, that maybe it's squeezed close. So I'm going to try to open this here. Up. So maybe this works better, I don't know. Or it could be maybe that the particles are too heavy and are not able to go in. Well, this worked a bit better. So I can actually see it now go down again. And the rest of the water I leave up here. So again, yes, this was also again. One has to be a little bit careful when putting in a lot of water and then not putting on a cover glass. Do not, exactly, do not go up with the magnification too high. Otherwise you're going to rotate the objective into the water. This has already happened several times. Yeah, with me, it's not a big problem because my microscope objectives are of high quality and also watertight and can be cleaned relatively easily, therefore, but not all microscope objectives are like that. So we see that there are a lot of particles in here now. Okay, so I will now remove the other. I'm going to switch to microscopes again. So let's remove this here. And without a cover glass now. And I have to go down to the lowest magnification. This is not going to be the, again, the proof of concept. It's too bright. Here we go. Okay. And this is basically what we see now. There are huge things floating around here. Huh? And now we have to find our tardigrade again, which is difficult because there's so many things moving around, obviously. Huh? So I don't know. I might have to go up a little bit with the magnification again, because then sometimes we see it better. Oh, look. Oh, look at this. Oh, I found a tardigrade. This one over here. Do you see this here? This is the empty skin of a tardigrade, because what they do is they molt. So they shed their skin, and this is just the skin of a tardigrade after it's molted, so to say. Huh? So we know that there are tardigrades around, but this one is not alive. It's just the skin. Yeah? So again, this is the origin. So I did not. This is not an enrichment culture. Okay, so this is not. Yeah, that's interesting. I don't know what it looks almost a little bit like a. I don't know what these things here are. The trichomes. What is this one? Is this also a rotifer up here? It is. Oh yeah, it's a rotifer as well. Yeah. So, and now we're just basically searching again. That's actually one of the fun things of this hobby a little bit is you get rewarded with patience. But for actually making nice pictures, so if you've already seen my tardigrade videos that I made before, you're going to see that the tardigrades are quite isolated, or other cells are sometimes quite isolated, and you do not see all of those particles around. And the reason is, and I often did not tell you that, but I'm going to tell you now, this is because I actually picked off the tardigrades using my pipette and transferred it into clean water, so that I do not see any particles around it, and so that the tardigrade does not cling to soil particles or hide behind them. Yeah? So this allows me to make videos that are a little bit of a better quality. Yeah? Just for your info, that sometimes if I just want to show you a big close-up shot of a tardigrade, I've picked it off. Yeah, or in a video that I'm going to release now, there are going to be two tardigrades next to each other. Okay, and they were not next to each other by coincidence. I put them together simply to have a nice video with two tardigrades next to each other. Yeah. So in that sense, you can say that sometimes I'm cheating a little bit. Yeah. But what you see right now here is this is how it looks in original. So there was a tardigrade all the way over to the left. Overlooked it. And I don't know. To the left. I don't know. You see, the problem is there is this delay when you post a comment. So first of, when I say something, then there is a several seconds delay until you're able to see it, and there's also a little bit of a delay. So that's a little bit the problem, that I'm not able to take the instructions completely in real time. But what you see over here, that is, for example, the so-called the test of an amoeba. A test is a shell of an amoeba. Sometimes you're able to see the pseudopodia, the arms extending outwards. Yeah. So let's have a look here on top again, worms. So, by the way, I just want to, before I forget, something that I also made in one of my previous videos, which I would like to recommend, because people want to know sometimes, okay, what is it that I'm seeing? What you can try to do is with a new version of ChatGPT, it actually does work. It is able to identify, or at least narrow down, the microorganisms that you see, provided that you've got a good picture. Okay, so if you are able to isolate the organism, and provided the organism has sufficient complexity, then it actually might be able to identify what it is, or at least narrow it down. So what about this one over here? Is there something here? The whole thing is moving here as well. So ChatGPT is an interesting thing. And once you've got some possible recommendations of what it could be, then you can actually do a Google picture search to verify. Again, here in the center, the test of an amoeba. They have these egg-shaped structures. Okay. Yes, commons, nematodes. Yeah. Okay, the tardigrade was kind of free-floating. So again, this is Lacrimaria with a long neck. I don't know. Lacrimaria actually is able to extend its neck very long. Is this? No, that's also a rotifer here. If I'm not able to find it, then I'm still going to try to illustrate a little bit of how. Yeah, again here that's the skin, not the skin, or rather maybe the exoskeleton, if you could say. Of a tardigrade. Yeah, and I'm quite sure that there are several in here, but it's easier to actually find them. When you look through the microscope itself. One of the reasons being that, of course, right now here on the video, the field of view is not quite as high. Yeah, here we're now at the border. Yep. Oh yeah. It doesn't matter. If I can't find one, I'll just choose any other organism. It's another rotifer here in the middle. Ooh, what is this? I have no idea what this is. It looks almost like also some kind of an insect. And maybe there's some parasites or some ciliates living in there, kind of feeding on it. I don't know. I think am I able to I'm afraid of actually going up with the magnification a little bit here, because I might otherwise touch the liquid, because again I'm not working with a cover glass now. This is if you look up here, that is 10 times. This is my 10 times objective. Yeah. But again, it might look different under your microscope, because it depends a little bit also on the magnification on the screen, right? And so if I were to look through the microscope here, of course I'm going to see a larger field of view. Yeah. That's kind of interesting here. Never seen this. Almost looks like that those cells in here. Yeah, it's inside some kind of a, I don't know, an arthropod. Maybe some kind of it looks like, counting the legs, some kind of a crustacean maybe. Maybe they're kind of feeding on it, you know. Yeah, that's that's the interesting thing here, right? So again over here, that's a rotifer. Yeah. Yeah, it's such a pity that I'm not able to find it now. But I know that we've seen it, that they are around. But again, it's a question of patience. Okay. You know what I'm gonna try? Where was this again? Here it is. I'm just gonna use this as a. I'm just gonna try it. Okay. I would like to look at these things here at a higher magnification. This means I have to put a cover glass on top. Okay. So what I'm gonna do is I'm going to try to pick it off. Okay, this thing here, right? So I assume that these are kind of single-celled, maybe protists, I guess, in here that are kind of feeding on this dead crustacean here, right? And I would like to have a closer look, so I'm just going to try to take it off using my pipette, okay? Instead of a tardigrade. Might not work, okay? So this is just a demonstration, just to show you what I've been doing. And sometimes it doesn't work, okay? Because sometimes I shake a little bit and I'm losing it, yeah? But I simply would like to illustrate to you how I would do that and how I have done this before. Okay, so I'm going to quickly switch over to the desk view here. Okay, I'm going to move this out of the way. Now I'm going to show you how I'm preparing those plastic tips. So I'm taking one of those, okay, just like before. And what I'm going to do now is I'm going to, if I want, I'm just going to use a microscope slide here to illustrate this. If there's the water drop now on here, and if I try to kind of suck it up using the tip, then I'm at an angle, okay? And if the water drop is not thick enough, then the front part of the tip will not be covered completely in water, and I will be sucking in air. Okay, so that's a little bit the problem. And I cannot go steeper because the microscope objective is in the way. Okay, so I need a steep angle, but I cannot get a steep angle. So what I'm doing is the following. Look, I'm just taking this tip and I'm just bending it like this. Okay, it's a disposable tip anyway, without actually getting air into it, right? And then it's much easier for me to actually pick up the sample, because at a steeper angle, the front part is completely underwater. Okay, and that's what I want. Okay, so sometimes I found this easier to pick up specimens this way. So that's simply an illustration. Yeah. So let's have a look. And again, because this is a little bit now some luck as well. Okay, so what I'll be doing is I'll be moving in. You can see it on the top. You can also see it on the side here, and I will be trying to stabilize the whole thing using a finger here. Okay, and this is going to work only if I go down with the magnification now to 4x, because I need the field of a view. Okay, I will be coming in, if I'm coming in from the bottom here, then on the microscope you should actually be seeing it coming in from the top. Okay, so this is a little bit, and now this is going to be a challenge. Okay, and I might move over now to my microscope, so this means my face is not going to be visible now. Okay, yeah. So, but I might be able to move at least the microphone over a little bit, so that I'm going to move so that when I'm working under the microscope, that at least you're able to listen to me. And I'm going to also move up here a little bit, and I'm going to try to look through my microscope as well. Okay, and let's give it a try. Okay, so I'm going to stabilize this here, and this is really going to be. And sometimes it also helps to actually rest the tip on the slide, simply for stability. So let's move forward. And I sucked it in now. Okay. Whew. Okay, maybe we were lucky. Okay. So I'm going to switch now over to the desk view. Here we go. And I'm going to now put it on here. So I'm going to make without cover glass, I'm going to now make a very quick check whether it's still on here. Okay. Let's see. Or not. Don't know. Ah, here it is. I found it. I don't know. Are you able to see it? Here it is. Okay. So let's zoom in more. Here we go. Ah, it's fine. And what I'm going to do now, okay, so what I'm going to do now is the following. I would like to add a cover glass. However, problem, the drop of water is so small that when I add a cover glass, it might actually crush it. Okay, so I don't want that either. Yeah, and for this reason, I'm going to add a little bit more water. And I'm going to do it like this. You know what? I'm just going to take the slide out. Okay. And I'm going to go to the desk again here. Because this is definitely too little water. And then the layer of water, I'm going to simply take it here. And the layer of water might be definitely too thin. So maybe this is better. And you have to separate the cover glasses. Dry wiping it as well. And up, hoping that it did not damage the specimen too much. Yeah. But scope. So let's give it another try. And let's try to find it. Of course, now it could be anywhere beneath it could be anywhere now beneath the slide, right? So that's a little bit the problem. So what we have to do now is we have to kind of systematically, more or less systematically scan everything. See, and that's always a problem when doing things like this. But I needed to do that because I really want to go up with the magnification, maybe even 60 times. And that's why usually, or sometimes it can be that processes like this actually will result in a loss of the specimen. No, I found it. Here it is. Very nice. Good. Here we go. Okay, so, and now without big problems, I can go up with the magnification. Okay. And let's have a closer look at what this actually is. Condenser. And here we go. Look at this. Never seen anything like this. And 40 times. Change the prism here as well. That is look, this is really weird. Let's make it a little bit brighter. I don't know. This is, in any case, this is not these cells here must be definitely feeding on the on whatever is in there. No, I don't think that these are. The question is that these are eggs, and I don't. I don't think so, because then the mother organism, the parent must be alive as well, but the parent is clearly dead. You only have an empty shell. Could it be that I don't know, we have some kind of a weird life cycle where the eggs kind of consume the parent? I mean, nature is so diverse, nothing would surprise me really. Yeah. Maybe he ate them first and then they grew. Also possible. Seriously, maybe these are some kind of parasites, you know? Yeah. This is fascinating stuff, honestly. What I'm going to do now, just to illustrate this a little bit, let's get the weird arrow out of the way again. Arrow, yeah. I want to show you now how the thing looks like in phase contrast, okay? Just for interest. So I have got a 40 times phase contrast objective as well. So this here is now not yet in phase contrast. It looks exactly the same as before, but I have to change now, something you cannot see. I have to change now the phase ring, and I have to open this up, and I have to remove the thing here, and this is now how it looks like in phase contrast. Okay. Yeah, actually, not so much better. Okay. But yeah, okay. I think, but you get the idea. Usually, phase contrast is good for observing bacteria. So actually, I was kind of thinking maybe there are a lot of bacteria in here as well, but not so many really. Yeah. So let's go back to the normal. Let's go back to the normal, and I'm going to now go up with the magnification yet further. I'm going to now use my 60 times. Boom. Now this is pretty nice as well. I have to go here with this here as well. And it's interesting, the effect is not very high for DIC in this case. Huh. Funny. Interesting stuff. Okay. Yeah. Yeah. I have a video of a huge bacterial colony in a dead tardigrade. Yeah. Yeah. So, yeah, these cells are interesting. I could indeed imagine that they kind of divided inside the body of the of this crustacean here, of this little tiny whatever it is, shrimp or whatever. Huh? Yeah, in case you don't know yet, I'm going to use this also for a little demonstration, because this 60 times objective, the one that I have up here, if you notice that there is a ring, look, there is it can be turned here, this, yeah, and this is for adjusting the thickness to the thickness of the cover glass. So by adjusting this, you actually are able to squeeze out a last bit of resolution from the sample. Yeah, so that is a little bit, but the effect is not so big now. So since they look unicellular, what are those dots? Fat. The question is those things that were, no, where is this? Those things inside here, right? What could it be? First of all, because, let's put it this way, because I'm able to see something in here, like those round circles, right? And because they're fairly large, I know that these are not bacteria. I'm just saying this. It's bacteria, those tiny dots that you see swirling around here on the outside, maybe. Yeah, these are bacteria. So these are definitely eukaryotes, right? So the globules that you see in here could be, I'm guessing now, maybe food vacuoles, right? Other cell organelles as well. Right. I do not think that these are photosynthetic, because otherwise they would be green. So that's another thing that I would assume here. Yeah. But indeed, it looks like interesting stuff. I'm also a little bit fascinated by this. Huh? So, you know what I'm going to do now is I'm going to go back to this here, to my original sample. Okay. And then that was the one where we were looking for those tardigrades, which we could not find yet. Yeah. What do you call the objective with a ring? This is simply a 60 times objective with a ring, with an adjustment ring for cover glass thickness. Yeah, it's a fairly expensive objective, and yeah, it's basically there to squeeze out the very last bit of resolution. However, I'm not really adjusting it very often. Yeah, so I guess it's useful for very specific purposes. Yeah, for very difficult to observe objects. Why does this? No wonder I have to adjust this here. Let me see. Yeah, I'm not getting a very strong color effect here for whatever reason. Okay. Just a second, or is it 5 times? Okay, no, let's just leave it here as is. And again, at the test of, yeah, these are again 2 amoeba tests. Let me show you again. This one over here and this one over here seems to be a dead nematode worm. Yeah, so basically, maybe as a little summary of oh, it's 1 hour 10 already. As a little summary, it's not only water samples. That's, I mean, of course, the whole thing here is in water, right? But I just wanted to make a point as well. It's also sometimes worth observing, moss samples, soil samples, and so on. This is also some interest. Again, I'm afraid I'm not using a cover glass now, so I'm afraid of going up with the magnification. Otherwise, I'm gonna get my objective wet. Yeah, a dead tardigrade somewhere. Yeah, or it could be again the skin of a tardigrade because they're molting. This means that they're shedding their skin as they grow. When the skin becomes too tight, the skin pops open and they're kind of coming out, and then they're a little bit bigger. And that's how they're growing. Tardigrades, by the way, they have an interesting biological concept. Oh, look, something's dividing here. It's called utily. This basically means that the adult tardigrades, have a fixed number of body cells. We humans, larger humans, bigger humans, have more body cells than smaller ones, right? But in the case of tardigrades, they have a fixed number. And if their size increases, then it's actually the cell size itself that goes up. So, and this is a concept called utily, when when they have a fixed number of cells. Some other microscopic animals also show this. Certain nematode worms also display the concept of utily. So again, here let's have a look. There's lots of movement going on here, possibly, or maybe some worms kind of moving everything around here. It happens that they may be trapped inside their exoskeleton, can't get out. Well, the thing is that those tardigrades, when they grow, this exoskeleton, they will actually pop open. And actually, now I'm a little bit lost as a biologist. Should we actually call this an exoskeleton? Because exoskeleton is actually something that insects and arthropods have. No, it's a rotifer. I think I might have already said it before. When they first found tardigrades back in the day, they were kind of debating a little bit, what is it? And they said it's, well, based on the number of legs, it could be related to the spiders, because tardigrades have 8 legs, and spiders as well. But of course, this is not the case. You know what I could do just as a last try? Maybe we're simply not lucky with this one over here. This could be, yeah. So maybe I'm just going to try another one. Okay. So I'm just going to use this slide here. Okay. And while you can watch this here, where is this? The desk view again. Oh yeah. So I need again a tip. Yes, tardigrades have their own phylum. That's correct. Yeah, they basically are a category on their own. So, okay, just a second. You want to see, of course, what I'm doing? And let's try this here. So I have to switch over to the microscope view. It's already in place. So and that's now this new sample. Yeah, and again, we have to look around. It does require a little bit of patience. Okay, well, unfortunately, let's give it a few more minutes. What time is it? It's 1:15. Yeah, in a couple of minutes, I'll simply quit again. I do have an organizational announcement to make. It's next week. I will not be doing a live stream just next week because I don't have time. So it will also not take place on Sunday because I'm not quite sure when I will return home on Sunday, and then it would kind of stress me out a little bit. Wow, look at this! What is this here? It looks also like yeah, and for this reason, I won't be doing a live stream next week. So you please don't wait. But I'll be uploading some videos, so you can at least look at those. Yeah, just for your info, because I don't like to be rushed, and otherwise I have to go home because the live stream will start, and this kind of stresses me. And then, for this reason, yeah. 91 people watching. Somebody tells me that's actually quite nice. So. Yeah, I know these some of you, some people who are watching might not be might have joined late. So what I'm doing right now is I'm looking at a sample from a moss. Okay, a sample that I collected a few days ago, and I'm currently looking for, it's a mite. I'm currently looking for some tardigrades, and I was lucky before. I found one before, but I could not pick it off. And I would like to find now one that I'm able to pick off. Huh? Otherwise, you know what I'm gonna do? Yeah, I'm just going to pick off this mite here. This shouldn't be too difficult. And maybe we can isolate this mite and get a better view of the mite, because there are so many other things around it. Okay, and maybe some of you might have not seen what I've done. So I'm going to now do the following. I'm going to try to pick off this little guy here, and again, look what I'm doing. Because these mites are pretty large, yeah, I have to essentially cut off the tip here so that it won't be. Oh, it's not pretty large. Okay, so that I'm able to pick it up. Yeah, it's fairly. The hole is quite okay. Yeah. So let's have again the scope view. I have to go down with the magnification. So here we go. So I want to pick off this guy here. Okay. So that's the one I want to take off. And I'm coming from the bottom. That means the pipette will be coming in from the top. So this means if it's coming in from the top, let's move it like this. Okay. So let's give it a try again. I'm going to move over again, okay, to my microscope, and I'm going to move this to the side as well, because otherwise it's kind of in my way and I don't have good access. So, and I'm going to now look through my microscope. I have to find it. Where is it? Where is it? Where is it? I cannot find it. What? Down here? Just a second. Oh, here it is. Okay, got it. So, okay. Here we go. I hope I'm able to suck it up. No, it didn't work. You know why it didn't work? It didn't work because the hole was so big that it wasn't completely covered by water. Okay, so I have to hold it a little bit, yeah, maybe at a higher angle, or I have to make the hole smaller. Nope, I'm not able to pick it off. Can't do it, won't work. I'm going to bend the whole thing a little yet further to go almost completely, yeah. And let's try it again. No, it's still here. Now it's right in the center. So I'm going to try something different. I'm going to try now. I'm not going to make the hole quite as big, but I do have to make it bigger. Otherwise, it's not able to pick up the tiny mite. So let's bend it again. Yes. No, it's still there. Here at the very top. Now in the center. Yeah. So let's try it again. And no. I think I might have it. I don't know. Okay, let's do the following. Let's try to release it here. And I think we've got it here. Here it is. Okay, and here we go. Okay, and this way I am, yeah. And now because it's in a cleaner water, for this reason, yeah, I'm able to make nicer videos. Okay, so that's a little bit the trick or the strategy that I'm using to get nicer videos, right? So what I could try now is I don't know, I could actually try to add a cover glass, but this might crush it again. Yeah, is it possible to catch Lacrimaria with a pipette? It was not far away from the mite. Okay. Yeah, I could try to do that if I find it again. Can you turn the mite into a permanent slide, or is that not possible? Yes, I can. You can turn it into a permanent slide. As a matter of fact, it's not even that difficult because the mite, and just like insects, I mean, mites are not insects, but just like insects, it's the same thing. They have an exoskeleton, a hard outer exoskeleton, which can be preserved quite well. So what I'd have to do here is I'd have to take the mite, I'd have to kind of maybe put it into alcohol, dehydrate it, and then to remove the water, and then I can mount it. Yeah, so that would be possible. And if you use Uperol as a mounting medium, and I've got a bottle over here, right? Then this mounting medium will actually also cause the exoskeleton to become more clear, right? I'm not going to do that now because it would take too much time, but actually that would work, making it into a permanent mount. Yeah. So yes, yeah. So I'm just quickly going through the whole thing here. Yeah. Yeah, mites are tricky. Smashed one with a cover glass. Yeah, that's generally the issue because larger specimens can be crushed quite easily. Yeah, so this is a little bit the thing. But I just wanted to show this to you how you can actually make slightly nicer specimens, so to say, by picking them off. And there are also pipettes out there that are yet smaller. I don't have one of those, but yeah. So, but let me check again. Here, this is the one that we had from before again. Where is it? No, this one, the one over here. That's the new sample. And I mean, maybe last look for some tardigrades. But you see, I mean, it's not so easy, right? And so, because some people have been looking for quite some time, are really happy when they find one. And even here, in this case, even though there are so many organisms around, it does take some time and patience to find them. Huh? So this one over here is not one. Is this? I was already wondering. Just a second. Again, here I have no cover glass. But have a look at this guy here. Let's see if it changes shape. It could be an amoeba. Yeah. This one here, right? I'd have to go up a little bit. I think I could do that. Let me check. I don't know if I'm able. No, that works. Okay, yes. Just barely. I don't know. It's numbered 20 times. Again, without cover glass. I don't know if it's an amoeba or not. Could be. Yeah, it does change its shape a little bit. Yeah. Yeah, those long strings that you see here could be bacteria. Yeah. There are some filamentous bacteria also around. I don't know what this is. Seems to be some kind of almost like a glass shard or some kind of a mineral piece. It's now using my 20 times objective. Yeah. Is this one? No, it's not one. It's not moving. Okay, people, I think... oh, look at this. Here it is. This lacrimaria. Ooh, how am I going to take this off? Ooh, should I try it? I don't know. These are difficult because they move around so quickly. Yeah. Are there prokaryotes that are not bacteria? Ooh, that's an advanced question. Archaea are prokaryotes that officially are not eubacteria or bacteria. Within the prokaryotes, these are single-celled organisms that do not have a nucleus. There are actually two categories. And actually both, they're called archaea and bacteria. You can also say that there are two different, or they used to be called archaebacteria and eubacteria. So essentially, in other words, the bacteria can be divided into two groups, and one of them is called bacteria. So that's a little bit the thing. So those archaea or archaebacteria are prokaryotes that live in extreme environments like volcanic areas, volcanic hot springs, acidic springs, and so on. And they have been discovered that there are these two types of prokaryotes by genetic analysis back in the 1970s, even already. Yeah? So actually in nature, there are three so-called domains. Yeah, and the domains are the eukaryotes. These are basically those organisms whose cells have a nucleus. Like, for example, we belong to those. All of the multicellular animals and plants belong to the eukaryotes. And then you have two more domains, the bacteria, just bacteria, right? And the archaea. And the archaea are essentially also just like the bacteria. They look like the bacteria on the outside, but are very different or somewhat different chemically, biochemically, and also from their DNA. Yeah? So that's a short question to a complex question. Yeah. Yes, that's correctly spelled. Okay, archaea, R-C-H-A-E. Yes, with A-E-A. Or you can also try archaebacteria. You'll find a lot of information about those. Yeah, it's actually one of the first things that you learn also in microbiology, that there are these two different domains of two different types of prokaryotes out there. So biodiversity is really enormous. And the interesting thing is why this is so remarkable is that those two prokaryotes, the archaea and the bacteria, are very, very genetically very distant from each other. They're as far away as we are away from the bacteria genetically. Yeah. So this kind of illustrates a little bit that just because objects look similar under the microscope does not necessarily mean that they're genetically also related. Yeah? So that's, I think, one of those interesting realizations, which is counterintuitive. I have never seen any multicellular prokaryotic organism before. Well, there are some examples, like for example, cyanobacteria, which are the blue-green algae as they used to be called, but they're actually bacteria. They appear in long chains, or can appear in long chains, and they can already show a first step into multicellularity. They're not officially multicellular, because bacteria are not multicellular, but they show a first step into multicellularity because they already show a very basic form of cell specialization. Because truly multicellular organisms are made of cells that are specialized. And those cyanobacteria or blue-green algae already show a few cells called heterocysts that are different from the other cells. So one might say that this is a first tiny step towards multicellularity, but actually they're not really multicellular. So you see that in nature is very, very diverse. Yeah. So that is a little bit. Okay, people, 1 hour, 30 minutes. This live stream has already been. Okay, and what I'm going to do is I'm just going to call it quits now, okay? I think it was, I hope you kind of enjoyed this live stream as well, yeah? So generally, I also do have another question, and maybe I'm going to start a poll a little bit, is that if you want me to, if you have any specific interests of what I should make videos, I mean, this is a very ad hoc type of live stream. I just basically put stuff under the microscope and we're going to see where it's going to take us. But if you have any specific interests, okay, or questions, then I might actually also do videos. So I'm always grateful for video recommendations. If you're more interested in the more technical aspects of microscopy, I do also have a second YouTube channel, which has a similar name. I call it Microbehunter Microscopy. This channel here is just called Microbehunter, but I've got another one called Microbehunter Microscopy, where I basically talk about optics, camera connections to a microscope, do a couple of product reviews, so less about observing and more about the actual hardware. Okay, so you might want to check out that channel as well if you're interested. Okay, this is also an interesting one. It also seems to be a rotifer. Rotifers all over the place. Yeah, and the last comment, just something that I also consider fascinating. Just a second, let me just illustrate this. Yeah, so this. Okay, I bumped it. Okay, this here is a rotifer. Okay, yeah, it's a multicellular animal. Yeah, so it has already cell specialization, and it's made. I read somewhere around 1,000 cells. And those other guys that you see floating around here, those ciliates, pretty much the same size, is made of one cell. Isn't it kind of fascinating that some of these ciliates have a similar size than the multicellular animals, microanimals? Yeah, so it actually shows how big even the cell size difference must be. Yeah. So this guy over here, yeah, is where's the arrow? Here it is, yeah. This guy, yeah, here is one cell, yeah, and this rotifer here is made of 1,000 cells. Yeah? So yeah, that's kind of fascinating, yeah. Okay, here's another rotifer, right? So it is quite well possible that some multicellular animals, some multicellular microanimals are actually smaller than some single-celled protists. Just from the size, just to illustrate a little bit about how diverse nature is. Yeah. So all of those guys that you see floating around here, these are all single-celled. Yeah. Yeah. This here. This looks almost a little bit like a small diatom. Yeah. Okay, folks, I'm now really getting carried away because I'm still so fascinated with this stuff here. And I'm going to call it quits now, okay? yeah, hope you liked it again. Hope that this kind of also motivates you a little bit to put some of the samples under the microscope. In this case, moss, and yeah, what do more cells give as an advantage? I think it's, oh gosh, thank you so much for those questions. Basically, okay, very quickly, if you're multicellular, you have emergent properties. Short answer to a complex question, multicellular organisms like we humans show emergent properties. And this basically means we as multicellular animals are able to do things that single-celled organisms are not able to do. I give you a short example of this. We humans are able to think because we are made of billions, our brain contains billions of nerve cells, right? And this ability to think is an emergent property that we are able to do, which single-celled organisms are not able to do because they're only made of one cell. So emergent properties are a result in many cells playing together. I'm able to talk because I've got vocal cords and a lung and so on, and that's only possible because I'm a multicellular animal, right? So this basically means that even though all living things perform the basic functions of life, nutrition, reproduction, whatever, yeah, energy, excretion, homeostasis, there are a whole bunch of things that all living things have the same, but multicellular animals can do more than that because the cells are specialized and they show therefore emergent properties. We're going deep into biology now. Okay, but thank you for the questions, because maybe indeed that's the topic of a completely new video. Okay, and I'm not really gonna quit. Yeah, and I wish you all the best. Okay, and yeah, happy microbe hunting as always, and not next week, okay, but see you around again hopefully in 2 weeks time. All the best, and bye bye.